Good afternoon, everybody. It's a pleasure to be here today. My name is Iñgo Rodriguez. I am the team leader in biophysical analysis in SGS MScan in Wokingham in the UK. And the challenge for me this afternoon is just to actually throw you 29 slides in 20 minutes. But you are going to be with me through that, and I hope that we will actually just do it on time. Now, seriously, the challenge that we are facing these days is awareness, isn't it? If I have to throw you an actual reflective question, and I will say, what are we doing here? And I answer that question, the answer will be that sure that we are doing something else that actually making contacts, isn't it? Surely we are doing something else that identifying opportunities and learning. So what we are doing today here is changing in many ways our industry, in the same way that 10 years ago, industry was changed by other people like us doing the work that we are doing at the very same moment. If I am giving you this introduction, because I think that is relevant, is because I want to put that in the context of the techniques that I am going to present you now. If I would have given you this presentation 15 years ago, you would have thought that I was coming from an academic environment, not knowing what is the relevance of what needs to be tested in our industry for characterizing biopharmaceuticals. But things have moved quite along in 15 years. And that is the message that I want to pass you before I actually engage with my presentation. Because there are going to be some techniques there that can actually look a bit esoteric. They are not. They won't be in 10 years' time. They won't be in 5 years' time. Let's have that in mind. Let's face it. Industry is moving. We need to move with that speed, and we need to accumulate and actually adhere to what is coming for the purpose of actually just creating a more competitive industry, for the purpose of actually just promoting safer drugs, and as well for the purpose of actually just increasing our profits, because at the end of the day, this is a business. The title of my presentation is Increased Confidence in Formulation Design, based on orthogonal methods. I mean, I'm talking about preformulation. You will understand with me that then there is a very, very high relevance in actually just talking about biophysical methods. The program that I am going to do, just giving you a brief agenda, is going to do a very quick introduction of biophysical analysis. What does it entitle? What needs to be done? What is the current perspective in terms of the regulators? What is the regulators' view? Where does it fit? And some biophysical techniques of interest. I'm going to give you some study cases so you can see some real data in order to appreciate what kind of data we do generate in SGS scanning Wokingham in the UK. Biophysical analysis, what that entitle, is a very broad term and actually just come with quite a significant amount of different techniques that basically they are all of them just focusing in something that is very relevant for characterizing a biopharmaceutical. Protein structure, protein dynamics, and hydrodynamic properties is mainly what you are actually evaluating when you are using biophysical analysis. They are mainly spectroscopic, thermodynamic, and hydrodynamic techniques. And basically, what we are focusing is in higher order structure. What is higher order structure? We are talking here about not primary sequence, forget about that. We are talking about secondary structure, tertiary structure, and quaternary structure. All the biophysical methods I'm going to be talking about are actually encircled in those three levels of organizations of proteins. Why it needs to be done? Well, we know that we are working with very, very, very fragile systems. They are extremely complex, and there is a lot of parameters that, for much that we control, are going to have an impact in the characteristics, in the physicochemical characteristics of the material that we are producing, that we are analyzing. Temperature, freezing and thawing, agitation, stress, solvent surface effects, protein concentration. I mean, when we are changing a buffer, on purpose or not, we are trying just to actually understand how the materials react to that change in conditions. There is when it comes biophysical analysis to actually allow you to monitor those changes. Aggregation is a very hot topic in our industry. It was 5 years ago, it will be in 10 years' time. We still need to be looking for new emerging technologies that allow us to quantify and identify low levels of aggregation. There are a very broad range of different types of aggregations. It's a very complex subject, very difficult to analyze, very difficult to understand, and very difficult to predict as well. So the methods that you use for actually monitoring that aggregation has to be the right methods. And more importantly, it has to be methods that actually can be used in industry. There is no point in bringing with an alternative for actually just monitoring aggregation that doesn't fit with the requirements of industry. We know that. Why it needs to be done? Well, this is very important. Product knowledge, well-characterized molecules, quality by design initiative. We really need to comply with that and stick to that. It's a requirement. It's actual our vector in order just to follow, in order to characterize biopharmaceuticals. It does really support understanding to batch consistency. It's required for comparability as the guidelines ICH Q5E. It can be crucial as well to approval on follow-on biologics. And higher order structure analysis doesn't replace biosize in the same way that biosize doesn't apply higher order structure analysis. I think that they just go in parallel, isn't it? In order to broaden the understanding of your molecule. And this is very important. They are fantastic tools for rationalize the changes that you observe in your materials when they are being subject to different matrices of formulations. The guidelines, and this is a very, very important topic. What the guidelines say about biophysical analysis. Well, not too much, unfortunately. Where you go to the ICH topic Q6B, that is the bread and butter of our industry in regards to what you need to actually contemplate for characterizing a biopharmaceutical, you will find that there is a list of methods and approaches that you need to evaluate in order to actually just comply with that guideline. But you will find that in the physicochemical properties, there is a little mention in regards to spectroscopy profiles. I think that that is an understatement. These guidelines were written in 1999. I mean, we are in 2013. It has passed a lot of time. Technology has advanced, isn't it? So we should need to accommodate to the new requirements of our industry in order to actually try to redraw new guidelines. And if those guidelines are not redrawn, our responsibility is to actually just bring these technologies and complement what is written in the guidelines in order just to provide a further consolidation of our experimental approach. That is a very interesting report that I will advise if you are interested in reading, that actually just bring some biophysical analysis and put the techniques in consideration with one towards the other. Very important guidance for industry that came along 1.5 years ago by the FDA, in which one, this guidance for industry is at the moment a bit in the air, is floating around. I think that is under evaluation. But the emphasis has changed considerable in regards to what is required now for testing biosimilars. And what you will find in these guidelines is that there is a very, very, very direct quote in regards to actually characterizing your materials with biophysical methods. In order to check the higher order structure, there is direct quotes in regards to techniques like AUC that 10 years ago were considered academic techniques, has techniques that are considered now to be golden standards for cross-validation of site exclusion chromatography, direct quote from the regulators in regards to the use of biophysical analysis. Where does it fit? The reality is that if you follow this graph that actually gives you the analytical activity over years of manufacturing, you will actually realize that there is a peak there that starts in pre-formulation through accelerated stability trials and then decay gradually as you move towards GMP production, because the techniques are very, very sweet to be used at the early stage. I'm going to just go very briefly through this, just to name some of the techniques that you will see later on in the study cases. Techniques like circular dichroism, FTIR, Raman and UV resonance Raman spectroscopy, all techniques that actually report in the secondary and tertiary structure of biopharmaceuticals. Intrinsic fluorescence and extrinsic fluorescence, very, very sensitive techniques to use to identify minor changes in the tertiary structure. UVB second derivative, very easy to use. Another technique that you can actually put through in order just to use an orthogonal method for the elucidation of tertiary structure changes. And differential scanning calorimetry, real useful techniques that is used in order to identify changes in the thermal stability of biopharmaceuticals through the actual determination of the melting point. SBIUC, a very, very relevant technique these days in order to quantify aggregation and oligomerization. Techniques a bit more qualitative, but still very, very important, like dynamic light scattering, very friendly and very easy to use with great deal of sensitivity, but it comes with a problem that is a technique that is a bit qualitative. For that, the technique has to be used in an orthogonal manner. And then techniques like SECMALS that actually just are based in a standard site exclusion chromatography systems, but allow you to actually just directly determine your molecular weight. Case studies, some examples, SBIUC. Here what we go is an IgG in 3 different formulations. Yeah? Basically, what we are testing here is what is the impact on those formulations in the higher order structure of IgG. the first thing that we are going to test is the propensity for aggregation of those materials. What is the impact of those buffers in the aggregational profile? And basically what we are observing here is that we got a major peak there, followed by a second smaller sedimenting species that corresponds to a monomer and a dimer. SBAUC, as you can see, is able to resolve that dimer very clearly from the monomer contribution. And not only that, it's going to give you a number. It's going to give you a quantification. We did that in formulation A. We did that in formulation B and in formulation C. And in that table, basically what you got is that initially we were having a percentage of dimer of about a 15%. And through the design of that formulation, we were able to actually reduce that to about a 4% and to about a 1%. So it looks like formulation C is successful mitigating the amount of dimerization present in this material. The next question is, right, we know that formulation C appears to be successful. What is the impact of formulation C in the higher order structure at the secondary and tertiary level? And that is what we did these experiments. In the left panel, you got FTIR spectroscopy, ATR, attenuated total refractance experiments, in which when we are able just to do the second derivative baseline normalized ATR spectra, force it to overlap and calculate the area of overlap in order to try to identify secondary structural changes. We did the same with circular decroissing. In the upper panel, you see the typical spectra in the far UV following the secondary structure. In the lower panel, you see the tertiary structure, giving you information in regards to the optical activity of phenylalanine, tyrosine, and tryptophan residues. So now we are talking about tertiary structure. We are able as well to estimate the relative amounts of alpha helix and beta sheet from those materials using those techniques. We keep throwing more analytical techniques. In the left panel, what you see is those experiments reproduced in terms of steady-state fluorescence. This is the inherent fluorescence that comes from the tryptophan residues. What you see in the right panel is the overlapping of all those experiments, showing you that there are no differences in between those three formulations in terms of the tertiary structure that is localized to the tryptophan residues. Now, interesting, we throw some DSC. And basically what we got is for formulation A and B, melting points of about 73 degrees. Formulation C is showing an actual 72.02. Is that significant or not? Is that falling within the variability of the experiments? I know that it doesn't. I know that that is big enough, but it's borderline. What we do now, are those materials different? Is the thermal stability changing? CD and FTIR say that nothing is happening, but we know by SBAUC that the dimerization content has been reduced. DSC is telling us that there is a correlation there. Let's keep throwing more techniques. There is when we come to a steady state extrinsic fluorescence. Sorry, that is a mistake in the title. Basically, what we use here is a dye that responds to the hydrophobic surface of the protein. So you got a conformational change, and you got hydrophobic residues that are exposed in the surface of the protein. ANS, this ANS is going to actually stick to those hydrophobic patches, and it's going to fluoresce more. That is the theory behind. Basically, what we see here is that in those three upper panels, what we see is the data that we are obtaining by fluorescence with ANS, and what we see in the right lower panel is a representation of the linear response of ANS as a function of concentration of protein. And what you can identify is that formulation C got a slightly different slope. Formulation AAB appears to actually just correlate very well. What this is indicating is that looks like formulation C is having more hydrophobic residues exposed in the surface of the protein. This appears to correlate with the DSC measurements. Something is going on with formulation C. Although it's able to mitigate the dimerization process, some structural changes are present at the tertiary structural level. So this prompts an investigation. Right, what is the significance of those changes? Let's do an stability, a short, small-scale stability study under refrigerated conditions. And what we did find is that formulation A and B, nothing changed over incubation at 2 to 8 degrees for 45 days. But what we did find when we were incubating formulation C at 2 to 8 degrees for 45 days is that we were losing the secondary structure and the tertiary structure. The left panel is showing you the CD data. Big changes in the secondary structure are associated with the incubation period. We see the same in the tertiary structure. Obviously, if you will see changes in the secondary structure, the tertiary structure has to be compromised as well. What we see in the fluorescence data is a very strong correlation with the CD data is actually indicating. As the actual incubation progresses, what we see is the fluorescence emission spectra increases in quantum yield, and at the same time, has an associated blue shift. That is indicative of those tryptophan residues being in a more hydrophobic environment. So something is going on with the tertiary structure of the materials. In order to top up those observations and actually just bring some orthogonal methods, we actually did some tests with ultraviolet resonance Raman spectroscopy, a technique that is able to identify the actual conditions of hydrophobicity in the tryptophan environment. And basically what we were observing here as well is that when we were incubating the material in formulation C, what we were observing is a change in the intensity of the ultraviolet resonance Raman bands. When you do the subtraction in between those spectra, you actually get an increase in the Raman intensity of the tryptophan residues. That is telling you that the hydrophobicity associated to those tryptophan residues is increased when you are comparing T0 against T15 days. You actually do the same experiment when you actually go to T14 days, T45 days, you not only see that the tryptophan environment has been compromised, but you see massive changes as well in the intensity of the tyrosine bands. So this is indicating that the tertiary structure is further collapsing. A very rich and informative technique to use in order to monitor the tertiary structure. We throw some AUC as well in order just to try to determine what was happening from the point of view of the aggregation of the materials. And what we do observe is that that is something that you don't want to see in AUC. After 30 and 45 days, we are having a lot of aggregation, high molecular weight aggregation associated to the materials in formulation C. Dynamic-like scattering is picking up that. This is dynamic-like scattering data accumulated in the material at 45 days, showing you something that is indicative. This is a volume distribution showing you that you got high molecular weight aggregation present in the materials. So all in all, the results are actually just putting together to give you an actual appreciation of what the materials are suffering from. If you actually pick up just one single technique and you actually just put all these experiments together, you are going to have just an angle of it. There is a need for actually just use orthogonal methods for biophysical analysis in order to create a scenario that you can rationalize, that you can understand. A very, very quick case study just showing you some of the dynamics of this data. This is cytochrome C. Cytochrome C can be unfolded at low pH. It suffers from an acidic unfolding. It's a typical model compound to use for research. And it actually shows very nice data. We see the spectroscopy. What we see here is that we go from pH 7 to pH 2. And we see a sigmoidal curve that is actually indicative of an unfolding cooperative process undergoing cytochrome C when we change the pH. What we see is that from pH 7 to pH 4, nothing appears to happen. Yeah? From the secondary structural point of view, we follow the tertiary structure, and obviously what we got is the inverse sigmoidal function. Everything is fine. Now, we did some DSC, and, well, we got some news here, because basically from pH 7 to pH 4, we see differences in the TM. Yeah? So the thermal stability of the materials, as you reduce the pH, is being compromised. From 84 degrees, very stable material, we know that from cytochrome C, to almost 77. So we got 7 degrees difference here from going to pH 7 to pH 4. And if I go back from pH 7 to pH 4, there are no difference by CD. Yeah? So how we put this together? It looks like there is a contradiction of data. Well, the reality is that when you are using these analytical techniques, you always have to have in mind that all of them are coming with limitations in regards to what they are actually just telling you. And those limitations are, in many ways, related with the capabilities of the techniques. Some experiments that I am undergoing at the moment with cytochrome C using ANS are actually showing me that there are differences in between going to pH 7 to pH 4. What happened is that those changes are very subtle, are actually reported by DSC, but are not being reported by techniques like CD spectroscopy. And this reinforces the need of use orthogonal methods in order to have a clear picture about what the materials are suffering from. When you are doing a comparability exercise, this is critical. You really need to know what your techniques are reporting on, and you really need to use orthogonal methods in order to gather enough evidence of any potential structural change. Last study case, very briefly, this is alpha amylase Bacillus subtilis. The material is mainly monomeric, but there is a tendency of these materials to actually just form electrostatic dimerization. And what happened with that electrostatic dimerization is that if you allow the material in a buffer that doesn't mitigate that dimerization has a function of time that is electrostatic, it becomes covalent. Because happens that the point of contact in between two monomers of alpha amylase are exactly where a cysteine residue that is very reactive happens to be. So this is a perfect example of how if you let a material to rest in a buffer in which one electrostatic interaction is actually being happening at that stage, that can become covalent. One is covalent, it's irreversible, there is nothing that you can do. So whilst it's electrostatic, you got the opportunity to actually establish a strategy for pre-formulation in order to mitigate that level of dimerization. Some DLS here, we don't see any dimer because DLS is a poor technique in order to report dimerization, oligomers. SDS page, nothing to see there. You throw some site exclusion chromatography and AUC, and we start to see things. Site exclusion chromatography, HPLC, is showing a level of dimerization of about 2% in the materials. And when you actually just test that against SBAUC, you can actually see that that correlates very well with site exclusion chromatography. The resolution is much higher in SBAUC, I have to say. And this is an example about how you can actually, once you identify these processes going on in a biopharmaceutical, you can actually do something about them when you got the chance to do it, when those interactions are electrostatics. These are experiments that has been done with SBAUC, has a function of sodium chloride, actually has a screening salt event in which one you are actually just mitigating those electrostatic interactions. And what we see here is that in the absence of sodium chloride, you increase the concentration of alpha amylase, and you actually got a concentration dependence in the dimerization of alpha amylase. So there is an actual correlation in between the amount of dimerization and the concentration of alpha amylase. If you actually just present sodium chloride in that solution, what you will see is that as you increase the concentration of the alpha amylase, you got a completely different behavior because in many ways you are canceling those protein-protein interactions. This is an example of how you can use, for example, a technique like AUC in order to clearly differentiate and characterize a process so complex like oligomerization. Key messages that I want to actually just leave in this room today is that they are very versatile techniques and report on key physicochemical parameters. And these physicochemical parameters might not be actually reported in the current ICH guidelines, but they will be in the future. Key applications are in preformulation, formulation, and force degradation studies, as we have seen through the case studies. What we actually are observing is a permeation of biophysical analysis through the actual industry. The techniques are becoming more relevant, and this has, in many ways, happens parallel to the advancement of biosimilars, in which an actual technical comparability exercise is needed. So the techniques are actually suffering from a revival. They have become very popular, and they are permeating through all industry. That is what I have just said, and this is very important. You need to use them in an orthogonal manner in order to actually just try to bring an actual rational explanation of your results. The perception in industry is changing. There is nothing that we can do about that. This is going to become more and more relevant in our industry. It's going to become more and more important, and our responsibility is to select those methods that we think that can be industrialized and are adequate for the requirements of the industry. And that's the end of my presentation. I hope that has been instructive and informative. Thank you very much for your attention.
I was just thinking, if you're using all these additional techniques, but the material is so precious when you're doing a formulation study, how do you cope with the low material requirements?
That's a very good question, because most biophysical methods are very, very demanding in terms of the amount of material that you need to actually just allocate for getting good data. The first thought that comes to my mind is the use of new technologies that are emerging. There is a response from the manufacturers of instrumentations to the demand of the industry. And there are clear examples of instrumentation that has been developed in the last 4 to 5 years that actually accommodate to low volume and low concentration requirements. You can actually identify DSC instruments now that are high throughput, that consumes very little amount of material, and they can actually have a turnover of 90 samples per day. There is advances in typical example is FTIR spectroscopy. FTIR spectroscopy, when I did my degree, I remember that I need to use about 40 milligrams per milliliter concentration in order to get a successful experiment. I couldn't do more than 3 experiments per day. And the actual uncertainties that I was having in those measurements were up to a 20%. Why? Because technology wasn't there at that stage. Today, you got manufacturers of FTIR instrumentation that they have actually identified that demand from the market. They have actually made the effort because they think that is what is going to be the vision and the scope of the industry in the coming 5 years. And there are fantastic instruments to these days that can go as low to 1 milligram per milliliter in concentration for FTIR spectroscopy, using volumes as low as 10, 15 microliters. And you can do up to 35, 40 samples per day. So the vision, the panorama, the landscape is changing constantly. And we need to be aware of that because there is a demand, there is a response. And that response comes from us and comes as well from the developers of instrumentation. And I think that it's very important for us to all be aware that all these biophysical methods that they were not used in the past because they were very demanding in terms of concentration and volumes at early development, these days you don't have that justification anymore. There is no justification for not using them. Well, thank you very much for your attention again.